Inside each of our brains, there are roughly 100 billion highly specialized cells called neurons that make about 500 trillion connections called synapses. These unique cells transmit important information, allowing us to sense and interact with the world around us. If you were to take a closer look, you would see that this information is transmitted between neurons using chemicals called neurotransmitters, where tiny structures called synaptic vesicles fuse with the membrane of one neuron and release chemical signals into the gap, where the second neuron can receive them. Scientists already knew some about how this neurotransmission process works, but now, thanks in part to over 10 years of collaborative research between Stanford University and SLAC National Accelerator Laboratory, along with the ultra-bright X-rays of SLAC's Stanford Synchrotron Radiation Light Source and LINAC Coherent Light Source, scientists now have a better idea of exactly how these tiny vesicles might fuse with the membrane of one neuron to transmit their signals. The key to this fusion is a cooperation between special proteins, called snares and synaptotagmin 1, that are then triggered by calcium to cause the vesicle to fuse with the membrane of the neuron. When a synaptic vesicle comes close enough to the membrane, the proteins connect with the two and enter a pre-fusion state. Next, when the neuron fires, calcium arrives and triggers the proteins, which bend the neuronal membrane toward the vesicle membrane and draw the two together. This finally triggers fusion, allowing the neurotransmitters to leave the neuron. This experiment represents the first time that scientists have seen how synaptotagmin 1 interacts with the snares at the atomic scale, and scientists are now also more confident that this protein group is assembled before calcium arrives, allowing the fusion process, and resulting neurotransmission, to happen very quickly, getting information from point A to point B in less than a millisecond. The end result is that our nervous system can work at incredible speeds, enabling us to sense, react to, and interact with the world around us. And now that scientists have been able to use the bright X-rays of SSRL, the LCLS, and the ANL-APS light source to see how this particular process works, it opens the door to better understand our nervous system in ways that even our brains can't yet think of.